Yes, sir.
Is it moving? Slides are moving?
No, sir.
No, slides are not moving. Same old story. Now slides are moving?
Yes, sir.
All right. Okay. We'll start. Shall we start? Hello?
Yes, sir. Yes, sir.
Can you hear me?
Yes, sir. We can hear you, sir.
Right, yeah. Okay, last class we were discussing factors that affect the engine activity. So we have seen substrate specificity, pH, temperature and engine concentration, substrate concentration, all those things we have seen. Those also affect the engine activity somehow, and other molecules apart from the environment, other molecules, biological molecules, those are present inside the cell. So they also interact with the enzymes, and then they may affect enzymatic activity or enzyme activity to increase its levels of activity that we call as enhancers, and some molecules decrease enzymatic activity, those called we can call as inhibitors or suppressors. Okay, so inhibitors slow down the rate of a reaction. So when you add the inhibitors, so some inhibitors, if they interact with the enzyme and enzyme changes its structure, then it loses its activity. So that's the way it reduces the reaction rate. Okay. Sometimes this is a necessary way to of making sure that the reaction does not proceed too fast. At other times, it is undesirable. So it is really very essential checkpoints in biological system. The enzyme should not keep on acting with their substrates. Okay, if they are keep on acting with their substrates, at one point substrates will be vanished in cell and the end product will be more inside the cell. So inside more end products are there. So when more end products are there, these end products may interact with other components, may cause undesirable reaction, undesirable effect. So these kind of checkpoints, inhibitors, are very essential in biological system. Okay, so you can see inhibitors, two types of inhibitors, inhibition activities we can find. One is called as irreversible inhibitors. Okay, there is no reversal of inhibition on decreasing the inhibitor concentration. So, for example, if you have inhibitor, the enzymatic activity is lessened. So if you decrease that inhibitor concentration, so enzymatic activity again will increase. Okay, so those kind of inhibitors different. Here, reversible inhibitors, once they add inside the enzymatic reaction, so once it is interacted with the enzyme activity or enzyme concentration, enzyme structural what is that structural formation will be disturbed. Okay, so it disturbs the bonds of that particular native enzyme and loses its activity. So this kind of inhibition we can call as irreversible inhibitors, like cyanides, okay, and penicillin and bacterial peptidases. Cyanides, particularly by covalently binding mitochondrial cytochrome oxidase, it inhibits all the reaction associated with the electron transport chain. So that's why electron transport chain enzymes all will be they lose their activity because of inhibition of cytochrome oxidase. And penicillin, if you add penicillin to biological microbial system, they will interact with the peptidases enzymes, and these enzymes loses their conformational changes, loses their structure. That's why they lose their activity. So this kind of activity inhibition we can call as irreversible inhibition. Okay, and also you can put a high denaturing temperature. Okay, where the protein completely denatured, it cannot be renatured. So that kind of in that kind of modification also you can say inhibitor. You can say irreversible denaturation. Okay, so inhibitors here molecules will be there. These chemical molecules are biological molecules. They will interact with the proteins and they may changes the structure of the protein. That structure cannot be revert back. Okay, so then the enzyme loses its activity. So you will see activity again from that protein. Okay, so this kind of inhibition we can call as irreversible inhibitors, and other type of inhibitors, second type of inhibitors, it is called as reversible inhibitors. Okay, the effect of inhibitor can be reversed by decreasing concentration of inhibitors. Okay, so here these are the competitive inhibitors. If you increase inhibitors, you lose the enzymatic activity. If you decrease the inhibitor concentration, you gain again enzymatic activity in reaction. Okay, so in this reversible inhibitors, again you can see competitive inhibitors and non-competitive inhibitors. Okay, so here mixed inhibition, just to understand this one, enzymatic reaction, you can see enzyme interact with the substrate, you can form enzyme substrate complex, and then you can get, if it proceed further, you can get product. Okay, so if you see the second reaction, Ki, which is enzyme interact with inhibitor, it forms enzyme inhibitor complex. Okay, if you add a substrate again, so enzyme substrate plus inhibitor complex will form. So that also, because of inhibitor is there in the active site, or inhibitor is there in somewhere in the binding site of the protein, loses its structure. So this complex will not proceed to go get the product. Okay, if you see the third reaction, enzyme interact with the substrate and enzyme substrate complex is formed. And if you add once enzyme substrate, once you add inhibitor, if inhibitor binds to that protein complex or protein complex, so it forms enzyme inhibitor substrate complex. So that reaction also, it is not possible to get a product. So first reaction, it is always possible to get a product because there is no molecule, there is no binding other than substrate molecule. There is no inhibitor molecules or interacting molecule, so that product you will get the reaction proceed to get the product. And second reaction, if you add enzyme plus inhibitor, there is also no enzyme active form, so even you add substrate, it will not get the product. If you add the enzyme inhibitor after adding the substrate, if the substrate is binding to some other part of the enzyme, so that changes the structural conformation, structural changes of that reaction, structural changes of that protein, so that also you will not get any product. Okay, so these are all the positive reactions, possible reactions in an inhibitor present reactions. Okay, so now we'll see competitive inhibitors. These competitive inhibitors, if you see the A panel is enzyme interact with the substrate, and substrate where it interact with the active site. Okay, you know that active site is the binding pocket or binding cleft of the substrate where the structure complementary to the active site. Okay, so when substrate interact with the enzyme, you can see enzyme substrate complex is formed. So enzyme substrate complex is formed. Once this proceed further, it gives a product reaction. Okay, so when you see next reaction, B reaction, inhibitor is also having the same kind of complementary structure to active site, and it looks like substrate molecule. So here, this substrate will compete with the substrate, this inhibitor will compete with the substrate at binding site. So if more inhibitor bind to the substrate, there is no place to bind for the substrate molecule. So the reaction will be reduced. The reaction will be zero level. So reaction will be blocked. Okay. So it goes and binds to active site so that the true substrate cannot get into binding at the site. Okay, so reaction stop or reaction reduced. You will see reduction in the reaction or reduction in the substrate formation. Okay, sorry, product formation. Okay, so if you see this kind of reactions, we can call as competitive reaction, where the inhibitor structure is very similar to substrate and it will compete to bind with the active site. Okay, so once it bind to active site, so if there is no binding site for the substrate, so reaction will not be as much as we predict. Okay, so for example, if you take arabinose competes with the glucose for the active site on glucose oxidase enzyme. So glucose oxidase is an enzyme, so it interacts with the glucose and it reduces the fructose molecule. Okay, so this glucose arabinose looks like very similar structure. So when you add arabinose compete with the glucose and bind to glucose oxidase reaction, and then it will not give any space or it will not give any much space to interact with the glucose. So the reaction levels will go down. Okay, this is one good example. And in these competitive reactions, you can see enzyme substrate complex is formed, enzyme product is formed. Okay, enzyme inhibitor, it is competitive inhibitor, so it will bind to all competitive binding sites, complete all binding sites, so form enzyme inhibitor complex, so it will not go further to get product. Okay, so here you can see one thing, the Vmax is unaffected. So when you plot the reaction, substrate concentration versus velocity, how much velocity, Vmax, maximum velocity, so you will see Vmax will not get affected in this competitive reactor, whereas Km is constant reaction, mechanism maintain constant reaction, that is has some value here. So the Vmax on Km will be decreased. So from here to here it is decreased. Okay. So in competitive reaction, competitive inhibitors, you need to mention that Vmax is, you need to understand, you need to keep in mind Vmax is unchanged, whereas Km is increased. Okay. So example for competitive inhibitors, another one more example we have seen arabinose for glucose oxidase enzyme, and one more here 2,3-bisphosphoglycerate. Another example inhibits its own formation by binding by bisphosphoglycerate mutase. So metabolic regulation byproduct inhibition. So 2,3-bisphosphoglycerate is a byproduct here. So when 2,3-bisphosphoglycerate is more, this molecule will go and bind to bisphosphoglycerate mutase enzyme. Okay, this is mutase enzyme, isomerase enzyme. So this molecule will go and bind this enzyme and will inhibit the formation of 2,3-bisphosphoglycerate. Okay, this is one type of reaction we call as feedback inhibition. Okay, so the end product or byproducts of the reaction will interact with the enzymes, so that those enzymes will go down at active levels, so they will not form, they will not get more concentration, more byproduct here. Okay, and also malate succinate. In this malate is the competitive molecule, so it interact with the succinate dehydrogenase. So here you can see in TCA cycle, you can see this enzyme. So succinate dehydrogenase interact with succinate and the malate is formed. So structurally very similar, malate is to succinate. So that will interact with succinate dehydrogenase and inhibit the reaction. And sulfonamides are the byproduct. Sulfonamides are the molecules which contain sulfate molecules. Okay, widely used in medicine to limit the bacterial growth. So these are the drug molecules, antibacterial drug molecules. So those are interact with the growth, some parameters in bacterial, some enzymes bacterial, and they compete with the substrate molecules and block the those reactions. Due to those blocking, those blockage, the growth will be reduced. Bacterial growth will be reduced. So these molecules we can call as antibacterial molecule. Okay. So this is competitive inhibitors and non-competitive inhibitors. We have seen same reaction here. We are going to see the enzyme will have active site where binding pocket for the binding pocket for the enzyme, and it will also have one more site which is called as inhibitor site. Okay, or we can call as allosteric site. Allosteric site means other site. Okay, allosteric site. So when enzyme interact with the substrate binding site or substrate with its active site, so it forms as a product. Okay, so there is no problem. Okay, so when you add inhibitor, non-competitive inhibitors, so these kind of competitive inhibitors will not compete with the binding site, but they will bind other part of the enzyme, where other site of the enzyme, so inhibitor binding site of the enzyme, and it will interact with the inhibitor site. And due to this interaction, some conformational changes will happen here. Binding site, conformational site, where active site conformational changes. Due to this, even though the enzyme bind to active site, this will not lead to formation of product because of inactivity at the active site. Okay, so this type of reactions we can call as non-competitive reactions. In these non-competitive reactions, inhibitors will not compete with the binding sites, but they will interact with the other site. That interaction leads to some kind of conformational changes at the active site. So that physiochemical changes leads to get inactivity or get lose activity of that particular enzyme, even though it interact with the substrate molecule. Okay, so this kind of reaction you can see enzyme substrate form enzyme product, and enzyme inhibitor complex will form. If you add substrate, enzyme substrate inhibitor will form, but it will not go further to get into product. Okay, so here you can see if you plot this one, substrate versus maximum velocity, Km is not affected. They have same Km value, but Vmax is decreased. This is the uninhibited enzyme, so Vmax is decreased here. So you can see change in the Vmax. Okay. So here, example, heavy metals like mercury, lead, and some heavy metals, chromium, acts as a non-competitive inhibitor. They will go and bind to enzyme or protein part. Due to that, some changes happen in the binding sites. They break the disulfide bonds here. And monoamine oxidase inhibitors that are used as antidepressants. They are covalently react with the enzymes in liver and effectively remove it. There are many potent drug interactions with monoamine oxidase inhibitors. One of these is tyramine, a compound that present in red wine and aged cheese. Okay, amine oxidase inhibitors and tyramine blocks neurotransmitter reuptake in brain, so it causes hypertensive crisis. Okay, patients are still subject to hypertension for long as 2 weeks after discontinuing of this drug. Okay, so these are the side effects I want to mention here. So if they want to use antidepressant, people use antidepressant molecule inhibitors like tyramine and other inhibitors for this monoamine oxidase is the one that causes the depression. If you use these depression drugs, that causes some kind of side effects like blocking of neurotransmitter, which causes the hypertension in those patients. Okay, so their BP levels will increase here. Okay, so if you reduce or if you withdraw those drugs, even withdrawing those drugs also have some effect till another 2 or 3 weeks. It will continue. Okay, that kind of side effect will be seen. Okay, next uncompetitive inhibitors, like non-competitive inhibitors. So here also you can see enzyme substrate product will form even if you add enzyme substrate complex. When you add inhibitors, it forms enzyme substrate inhibitor complex, and because presence of inhibitor, it will not lead to go to further reaction to get product. Okay, So you can see in these unconjugated reactions, both will change here. Okay, Km is also decreased and Vmax also decrease. Okay, so both Km value and maximum velocity of engine will decrease in these unconjugated reactions. Okay, so lithium and phosphoinositide cycle, an example for non-competitive inhibitors and its pharmacological consequences. You can see this about this inhibitor more in this paper. Okay, next one is irreversible inhibitors. We have already discussed these molecules will interact with the protein and break the bonds, and it will not revert back even you remove that inhibitors. That kind of inhibitor we can call as irreversible inhibitor. Cyanide is best example. Irreversible inhibitors of cytochrome oxidase enzyme found in electron transport chain in respiration. So even you reduce the reaction, the damage is already happened. That leads to death of that particular cell. Okay, where cyanide is interacted with cytochrome oxidase. If you have more, cyanide, so that leads to all cells death, and that leads to system death, and that eventually leads to that animal death. That's why if you see those people who took cyanide, it is not at all possible to revert back, to remove that inhibitor to get into normal condition. Okay, that is the problem with the cyanide cases, death cases. Very little success in these cases to revert the death. To revert the damage by the cyanide. Okay, now this is about the inhibitors. Any doubt you have? Subhash, do you have any doubts?
No, sir.
Okay, so now I will go to other properties of enzymes, general properties of regulatory enzymes. So we are discussing about these molecules affecting enzymatic activity, and regulatory enzymes. There are some regulatory enzymes we need to know before we go into further metabolic reactions. Okay, so in metabolic reactions, most of the reactions are done by these proteins we call as enzymes. Okay, so when you see the metabolic action, so metabolic pathways, one molecule will convert into another molecule, that molecule will become a substrate to another enzyme, and that like that. Okay, finally during this process you will gain enzyme, you will gain energy, and that molecule will convert into either carbon dioxide or urea or water molecule to excrete by animal. Okay, so during this metabolic pathways, some enzymes have its specific characters. Those enzymes we can call as regulatory enzymes. Okay, so in the biochemical pathways that you will see soon by studying are composed of group of coordinated enzymes that perform specific metabolic process in these metabolic activities. Regulatory enzymes usually the enzymes that are rate limiting or committed step in a pathway, meaning that after this step, a particular reaction pathway will go to completion. Okay, so these regulatory enzymes in this pathway, some stages will have a checkpoint we can call as rate limiting reaction or committed reaction. So if you cross that rate limiting reaction or committed reaction, that reaction will eventually go for completion. Okay, that particular type of enzymes, that particular step we can call as a rate limiting or committed step, and those enzymes involved in these steps in these stages we can call as regulatory enzymes. Okay, so frequently regulatory enzymes are near the initial step in a pathway, or part of branch points or crossover points between pathways. So where a metabolic can be potentially converted into several products in a different pathways. So these metabolic regulatory enzymes normally at the initial stage, okay, are part of branch points or where the crossing over one pathway to another pathway so that one molecule can convert into different molecules. Okay, so here these enzymes, as we see regulatory enzymes, they will be initial points. So once it is very important to start the reaction. So to start the reaction, so these enzymes may be sometimes a rate limiting enzymes. Okay, in general, a cell needs to conserve energy, therefore costly. Biosynthetic reactions pathways will be operational unless a particular metabolite is required at a given time. So these metabolic reactions, as you know, cell need energy. So cell use the energy very carefully. Okay. So unless that reaction product is required for the purpose of cell activity, then only that cell will utilize the energy and start the energy that pathway, metabolic pathway, or that reaction pathway. Okay, so these what it called enzymes will be there. Those enzymes are rate limiting enzymes. So these enzymes will make a kind of checkpoint, make a kind of stop points where cell protect its energy or save its energy until it is very required, very much required. Okay, so recall that when acting as a catalyst, enzyme mediated reaction should be reversible. Okay, however, regulatory enzymes frequently catalyze the thermodynamically irreversible reactions. That is a large negative free energy, that is del minus delta G, greatly favors the formation of given metabolic product rather than reverse reactions. Okay, so most of the these metabolic regulatory enzymes, you can see irreversible reactions. Okay, so they catalyze thermodynamically irreversible reactions. Thus, regulation of enzymes activity usually at committed step of the pathway. It is critical for supplying and maintaining cellular metabolites and energy homeostasis. So, general mechanism, there are few, several, factors like control overall the quantities of enzymes or concentration of substrate at present, and alteration of catalytic efficiency of enzymes. So this you can see many stages. We have discussed many stages are there, many steps are there to control the enzymatic activity or to maintain the homeostasis at substrate concentration or product concentration inside the cell. Okay, you can see here we already seen substrate concentration is one of the step to control the reaction, and the inhibitors are one of the steps to control the reaction. These inhibitors may be by products or other compounds, you can see, and like amount of enzyme is also one type of reaction, and allosteric activation or inhibition, that we have seen that in non-competitive inhibitors. So activation, if this molecule binds to that binding site, other site, so that leads to more activation of reaction. So much more faster reaction product you will see. That is called as allosteric activation. That molecule binds, it leads to changes in active binding site, that leads to decrease in activity. So you will see less reaction product or no reaction product, we can call as allosteric inhibition. Okay, so this kind of activity steps also present inside the cell, and covalent modifications like phosphorylation, methylation, and all those things leads to activation as well as inactivation too, and modular protein binding and proteolytic cleavage. Proteolytic cleavage is good examples, chymotrypsin. trypsin or pepsin synthesized as precursor molecule, pre-chymotrypsin, pre-pepsin, trypsin, like that. Okay, so these molecules synthesized initially as inactive form. So once they are required, these molecules get into activated by cleavage or by activating by interacting with other molecule, with an enzyme. So this molecule will get activated and do their activity. Okay, so if you synthesize this molecule as active form, they start by interacting with substrate and cleave. So say for example, pepsin is a proteolytic enzyme. Okay, there is no differentiation, there is no specificity. So once pepsin is synthesized as active form, directly into active form, so it will start binding with all proteins, it destroy it. Okay, so to prevent that one, there are some checkpoints. They were synthesized as protein or zymogen protein, inactive form of the protein, those called as zymogens. So then once requirement is required, some other protein molecules, small molecules, will interact with that zymogens, and they cleave that protein into smaller peptide. So once this smaller peptide formation is there, this smaller peptide become active form, and then it interact with other proteins. Okay, that is called as proteolytic cleavage. So there are, you can see many mechanisms, many steps are there in regulating enzymatic activity inside the cell. Okay. So the overall synthesis and degradation of particular enzymes is also called as turnover number, is one way of regulating quantity of enzymes. So this is also one type of mechanism. So all proteins will synthesize and they will have lifespan. Okay, so once lifespan is over, they will take into apoptosis. So this kind of process turnover we can call as turnover number. Say for example, alkaline phosphatase, just example, I'm not sure right hour. Alkaline phosphatase is 9 hours. That means it synthesizes and it do its activity and then it degrades by 9 hours. So the turnover number is 9 hours. Okay, catalase maybe 16 hours. Okay, so catalase will be synthesized and do its activity and taken into degradation and get into degrade. So by 16 hours. So catalase turnover number is 16 hours, like that. So this is also one step or one type of process to control enzymes in biological system by degradation turnover number. Okay, and then some enzymes get synthesized by the transcriptional activity. Okay, translation activity at gene expression level, and then you can see some molecule enhance the gene transcription by binding their promoters. So the RNA polymerase will interact with the DNA, and then mRNA will be formed, and mRNA will convert into protein. So the protein will be taken into different stages, methylation, amidation, and like that. Post-translation modifications, you can see, you can call those called as post-translation modifications. So they in this process also, this process post-translation modification is also one type of checkpoints to regulate the enzymatic activity. Okay. And this I already told zymogens, they synthesize basically in active form. So once once their role is required inside the cell, so other enzymes will interact with these inactive forms, make them active. Okay, so proteases enzymes, particularly pepsin, trypsin, and chymotrypsin, are synthesized first as large inactive precursors. They are called as zymogens. Okay, so like pepsinogen, trypsinogen, and chymotrypsinogen, these are all, if you say this nomenclature, they are, you can say they are zymogens and inactive enzymes. If you say trypsin, pepsin, and chymotrypsin, those are activated enzymes. Okay. So here you can see how trypsinogen, chymotrypsinogen, and pepsinogen get into active form. So they will, interact and make them active form. By interacting this, they cut some kind of portion of from that inactive form. So the shorted protein gets into activation form. Okay. So allosteric sites, okay, allosteric proteins, allosteric enzymes, these are Greek words. Allos means other, stereos means shape. Okay, site. Okay, that means in Greek terminology, allosteric means other shape or other site. Okay, these other sites are the sites where the molecule, inhibitory molecules or individual molecule will interact with the proteins and can activate. Okay, this activation we can call as allosteric activators. If this interaction may leads to decrease the activation, that we can call as allosteric inhibitors. Okay, so these allosteric sites are the not active sites and other sites. Okay, feedback inhibition, as I said in previous 2,3-bisphosphoglycerate example, is the enzyme which interact with the mutase enzyme, bisphosphoglycerate mutase enzyme. This is the enzyme it converts into 2,3-bisphosphoglycerate. Okay, so this enzyme will get inhibited by this 2,3-bisphosphoglycerate if 2,3-bisphosphoglycerate is more inside the cell. So there is no need to synthesize. There is more substrate or product is there. Okay, so this kind of inhibition we can call as feedback inhibition. In this feedback inhibition, you can see enzyme convert A to B. Okay, sometimes B may act as a inhibitor to this enzyme. Okay, so this B converts into C converts into D converts into E. So this E is the end product. E may act as inhibitor for this enzyme too. Okay, so this inhibition we call as feedback inhibition. Okay, once E is more inside the cell, so there is no requirement to convert the substrate into B molecule, E molecule. So this kind of inhibition we can call as product inhibition or feedback inhibition. Okay, so that they will interact with the enzyme and change its structure, and so that the enzyme loses its activity. This activity we can call as feedback inhibition. This is one kind of regulation mechanism inside the cell for the regulation molecule. Okay, these are the properties. Allosteric enzymes differ from other enzymes that they are generally larger and in mass and are composed of multiple subunits containing active sites, regulatory molecules, binding sites. So when you see allosteric enzymes, when you compare with normal enzymes may not have some that kind of complexity. When you see these allosteric enzymes may very large proteins and subunit proteins, they will have active sites and they will have allosteric regulatory binding sites. Okay, so but same principle that govern binding of substrate to active site are similar for the allosteric molecules also to bind active sites as well as allosteric sites. That means they should have complementary binding sites. If you don't have complementary binding sites, they may not bind to the site, either allosteric site or what is that active site, substrate site. Okay. So in this allosteric activity, you can see cooperative allosteric in relation to multiple subunits. Enzyme changes the conformational one subunit leads to conformational changes to adjacent conformational changes. So for example, 4 peptides are joined together form as a protein. So one allosteric binding of that one subunit that leads to conformational changes because it is interacting with the other 3 subunits, that leads to conformational changes of 3 subunits also. Okay, that kind of allosteric effect we can call as cooperative allosteric effect. And homotropic regulation, when bind one molecule to multi-subunit enzyme, causes conformational shift that affect the binding site of the same molecule to another site of that enzyme. So here subunit 1 got the molecule binding site, that is the other site, allosteric molecule. So same conformational changes will happen in other subunit sites so that that molecule can bind, regulatory molecule can bind to other allosteric binding sites present in the protein. Okay, heterotropic regulation, when binding of one molecules of multiple subunit enzymes affect the binding site of different molecules onto the enzyme. So here one molecule bind to one subunit regulatory molecules, that changes conformational activity, that conformational changes of that particular subunits, other subunits. Those subunits also accept to binding other molecule than this regulatory molecule. Okay, other molecule. Let's say, for example, NAD is binding site for allosteric site. Okay, that binds to one subunit. So this binding leads to changes in other subunits where iron can also bind, if iron binding site is there, or glucose can also bind, or some other FAD also can bind. Okay, so that kind of changes you can find in this type of regulation, which is called as heterotropic regulation. Okay, so here, as I said, molecule, those enhance their activity by binding. Those are called as allosteric activators, that we can call as allosteric positive regulators, and those called inhibit the regulation of the molecule, that those called as reduce the activity. Those called as allosteric inhibitors or negative regulators, positive or negative regulators. Okay, so you can see Michaelis Menten. We are going to see this one, Michaelis Menten equation. So here, this is the molecule, this is the reaction sigmoidal curve where there is no activator or inhibitor. Okay, so if you see activator, if you add to the activator, so you can see a kind of more what is that, hyperbola thing. And when you add inhibitor, you can see still some type of sigmoidal curve here in this enzymes. Okay, Tmax and Km will not change, affect the allosteric enzymes character. So there are different models for these allosteric enzymes. Okay, these enzymes, when they find these enzymes, it was very surprised to see they are acting differently from the normal enzymes. Normal enzymes means where they interact with the substrate and then they give the product. Where they interact with the inhibitor, they reduce the activity. Some molecules, they apart from that active site, some molecules have other sites. They interact with other molecules, get into more activity of the protein, or get into reduce the activity of the protein without having inhibitor molecule. Okay, so they started working on this, and they worked on this in 1965. A paper by Monod, Wyman, and Chong-Ox reported and modeled the structure, predict a model, they given the model. So they said that these are the introduced simplest model with two conformational of enzyme designated R and T. Okay, R is active form. Okay, that is R is active form. That means it has some kind of activator. Okay, T is less active form. Okay, R and T. Okay, so they are called as R means relaxed, where protein has relaxed structure, where protein has relaxed conformation. Then it will have more binding sites for the other sites. They will interact with them, and then these molecules get into activated. And trot site, where is less reactive, so it is not getting into active form. Okay, in this model, subunits of all oligomers must be in the same state. Okay, the T state predominates in the absence of substrate. S binds more tighter to R than T. Okay, where because relaxed form, active form is there, so it will have more interaction. It interacts more with the substrate. That's why S binds more R than T. Okay, T predominates where substrate is not present, so there is no interaction is there. Okay. So later, this model quickly followed and extended treatment by extended their model, extended further work, and Coastland and his colleagues they given two conformational changes using the circular and square model. Okay, circle for R and square for circle for T, that is shorter and less reactive, and squares for R model. Okay, so that you will see predominant differences even in the model also. Okay, instead of both circles, they have both circles, so they call as R and T. So that gives some kind of confusion. So they simplified this one. Circles for T rate less active, squares for R more active. So it is easy to understand also. So T and R. So T will be have subunits will have together less form of active form. This can convert into a more active square form where it will bind to other allosteric site subactivators. So this can interact more with the substrate molecule. More product will be formed. Okay. So they have given schematic representation, binding sites, substrate binding sites, and substrate binding sites as well as allosteric binding sites, allosteric inhibitor sites. All those things are they represented here, and then they show that this is substrate binding site, and activated binding site is different, and substrate bind if interact with activator, it will interact more with substrate, and substrate, yeah, activator will form and it will give more product. Okay, if inhibitor is there, inhibitor will bind and it will not interact with that substrate. So you can see sigmoidal curve, more sigmoidal curve in inhibitor, hyperbola curve in activator. Okay, these are the model coastline and colleagues given for this uh, allosteric activators. Okay, and then enzymes can also get activated, controlled by, as I said, like covalent modification, phosphorylation, by covalent modifications, phosphorylation site. Okay, this you will see in later in metabolic activities. Phosphorylation leads to activation of the protein, and removal of the phosphate molecule leads to inactivation of the protein. Okay, sometimes phosphorylation leads to inactivation of the protein, and removal of this phosphate molecule leads to activation of the protein. So you will see this kind of activity in different pathways. We are going to see later in metabolic pathways. Okay. Other modification, prenylation, methylation, palmitylation, this covalent attachment to these proteins also leads to activation of the protein. Glycosylation is also one type of modification to get into activation of these proteins. Okay. This glycogen phosphorylase, we will see more about this in glycogen synthesis. Okay, just for example, I have given here. Just go through. That's all. Any doubts? Do we
No,
have
sir.
any doubt?
No, sir.
Okay, right. So shall I stop now?
Yes, sir.